talked about flagella and pili and fimbriae. Let's see what else. Biofilm, capsule formation, all of those ultrastructures as well. So we're continuing on with that. The next ones we have to deal with are called paracrystalline surface proteins. Also known as S layers. And think of this kind of like a three-dimensional array of proteins that kind of overlap. So a lot like Kevlar. The finer you can get that overlapping, the stronger these are. And so what this creates is basically a cell surface layer that adds a lot of protection. Once again, kind of a common factor. So anytime we think of protection, think about protection from antibodies, antibiotics, white blood cells, and these guys also can use it to help to protect them from the environment. And like I said, it's kind of this layer of Kevlar. Now, one thing that we've kind of alluded to, but we're going to come back and talk to you about them just a little bit more. These part lipid, part polysaccharides that we find. Once again, these are usually anchored in the membrane. And we normally associate these guys with gram-positive cell walls, or excuse me, gram-negative cell walls. Now, they in themselves are kind of weird in that when they're attached to the cell wall, they don't have a lot of, they don't increase the virulence of these gram-negatives very much. But if you take that gram-negative and you break it into pieces, and that lipopolysaccharide is floating around by itself in a piece, it then becomes what we call an endotoxin. And so that's kind of what we see it as. So during bacterial breakdown, in many different ways. So for instance, if we have a white blood cell that comes in, phagocytizes this big gram-negative and breaks it into pieces, when those pieces get free, they basically end up being what we call these endotoxins. So as part of the cell wall debris during bacterial breakdown, we call these guys endotoxins. So we'll come back and revisit this, just a little bit of difference between the two. Exotoxins are excreted with purpose. Okay, so they're secreted. Endotoxins are just chunks of cell debris during cell breakdown. And they're just kind of toxic. So there's definitely a difference between them. These guys have genes that turn on, the bacteria stays alive, they excrete this through the cell wall into the media, and off they go.
So the endotoxins, are they something that just happens to be toxic, or are they that way on purpose?
They're that way on purpose, indeed, so they're trying to elicit a response. So usually these endotoxins end up giving us what we call toxic shock syndrome. TSS. Most men aren't familiar with it. They will be in this class. Most women have ever read the pamphlet in the tampon box have seen the big hazards about TSS, right? Well, it's not that there's a problem. It's the breakdown of those cells in the vagina during menstruation, releases those endotoxins, and we get that toxic shock-like syndrome. But TSS is basically something we associate with all types of endotoxins. That's just probably the only place that you see. Then we have all sorts of storage units that are extracellular structures, but these guys are inside the prokaryotes. So we're going to call them intra for being inside. And these guys are basically little vacuoles or storage units. And most of the time, 90% of the time, they're utilized for energy. They have a couple of different synonyms that we call them. Most of the time, they're called inclusion bodies, or sometimes they're just called storage vacuoles, basically the same thing. And likewise, as I just mentioned, usually for energy, not all the time, but a good part of the time. So we'll start with a big list of examples of what we find in these storage units. One of my favorite. So we have this thing called PHB, polybeta hydroxybutyric acid. Big, wonderful name. It's very lipid-like, so it contains a lot of fatty acids. So we can use it as an energy source. All sorts of polyphosphates. You guys think back to your general chemistry. These are phosphates, or excuse me, phosphorus with 4 oxygens and a -2. And when we have lots of these together, we call them polyphosphates, and they can kind of form these covalent bonds between the two. But once again, basically just utilized as an energy source. And then likewise, we have other sorts of elements that end up creating multi-branched types of compounds. And sulfur is an example of this, and it creates what we call polysulfur units. And this is where we see either anywhere from 4 sulfurs to 8 sulfurs interacting to kind of form a sulfur ring. So many times we see these in microaerophilics or organisms that don't use organics. These would be chemolithotrophs that use other types of chemicals for their energy source. And then we have these strange sorts of things that really aren't used as an energy storage. Called magnetosomes. And as you can imagine, it's made of iron magnetite. Everyone loves iron magnetite. You ever seen Breaking Bad? That's how he gets into the storage unit. Get all of his diethylamines. Not that I'm an expert at making methyl types of speedy products, amphetamines, if you will. But, so iron magnetite. These have an Fe3O4 composition. You have probably seen them in Etch-a-Sketch. So that's the black dust we see. So they're very, very sensitive to various types of magnetic poles. So rather than using it as a way to an energy source. Rather, it allows these organisms to figure out more or less what the magnetic north is and to orient themselves based upon magnetic fields, not necessarily magnetic north all the time.
Yeah.
And we'll go into these when we talk about taxis. Usually it's using what's called magnetotaxis. Or magnetotaxis for singular. That's this ability to move around based upon the magnetic fields of the earth. We see it with bats and birds all the time. Well, bacteria can also do it. And the last intracellular structure we'll talk about, we've already kind of seen in lab. These are those spores. I'm going to go into just a slight bit more than we did before. But it'll help to recap the whole process. And there are really two different processes connected here. The act of creating spores, we call that sporulation. And then once those spores grow out back into a vegetative living state, we call that germination. So the real endospore formers we have to worry about are those endospore formers we find in the soil. And the ones that are most pathogenic come from the genera Bacillus and Clostridium. So an example of Bacillus would be Bacillus anthracus. Just on a side note, those anthrax spores, which are really toxic, and the anthrax toxin that's produced, 3 different ways you can get it. They're all really not very good for you. The most deadly way is when you inhale it. What you don't realize is you have all these little lesions in your lung from coughing and sneezing and smoking and all of that you've done to yourself. And so those spores can get into your bloodstream really quickly. About a 95% mortality rate. Not a very good chance you're going to make it unless they catch it really soon. The next way is, of course, if you ingest it, then you're looking at about 80–85% mortality. And then the last way is if you get it through a wound. We're talking about 70–75% mortality. So basically one of the most deadly out there. Many people have tried to think about weaponizing this as a bioterrorist weapon down the road. The other one, of course, is Clostridium. And there are a couple of different Clostridiums we have to worry about. One here in Wyoming we see all the time is Clostridium tetani. It causes tetanus. So these spores are hanging out on the rusty metal that you get jammed right into your bloodstream, turns vegetative, creates that nice toxin. This is a true exotoxin, tetanus toxin, causes that rigid paralysis. Do you guys know any other Clostridium that might be out there? One of my favorite. Clostridium botulinum causes botulism. Botulism is one of the most deadly diseases on earth. So, the botulinum toxin is one of the most toxic toxins we can find, except for a couple of those weird sea snails that live on the ocean floor that are super, super toxic. So, usually we get this from things like weirdly prepared food, TV dinners, cans of food that have been punctured, and have been more or less with something metal, and we get those botulinum spores in there.
What do you mean weirdly prepared?
What's that?
What do you mean weirdly?
Like you have a lot of pre-prepared dinners, like pre-prepared and frozen, or pre-prepared and microwaved, or however. So those that are in cans usually, or sealed in weird vacuum bags, all depends. So we can get it in the food product a few different ways. Usually it's because there's a leakage in the container. This botulism toxin, if we were to take one Clostridium botulinum, stick it in a broth and just let it grow overnight, just the broth it's living in, we you take 1 milliliter of that broth, that's a very small amount. Remember how much we were using in the transfers? 1 milliliter can kill 2 million mice. And that's just of the broth. And you haven't purified this toxin. This is just what it's excreting out in its broth. So when you get botulism, you die. There isn't, unless they catch it and they know exactly what you have, but into the hospitals in this town, we know what that's like. Not that I have anything against the hospitals in this town. I think I've been escorted out of just about each and every one at some point. But I'm not too good with Western medicine and the whole practice of emergency medicine. But yeah, so really, really lethal. So these are things that we have to worry about when we're dealing with these spores. Once again, remember these are not for reproduction. Bacteria are asexual. They don't use these as gametes, but rather, this is a response to adverse environmental conditions. So that would be like lack of nutrients, UV radiation, various types of heat and pressure, changes in pH, acidity or basicity, conditions. Try that one again. So they think they're going to die, and this is just a last-ditch effort to try to protect themselves or their lineage before they conk out, if you will. So all of the energy goes into the cell to make these, and the cell is going to eventually rupture and be done with. So for sporulation, we won't go into each and every one of the steps, but it is a 7-stage process. And it usually takes somewhere around 48 to 72 hours for this organism to start and completely go through the whole process, so it takes a little bit of time. And eventually what we get is lysis of the cell and release. Now, we've talked about the morphology of this bad boy, so if you take a look at the morphology, it has this really, really tenacious outer exosporium coat. Inside of this, we have what's called the spore coat proteins, which is right on the inside. I think we got just about everything. So this exosporium coat, super tenacious, kind of like those S layers. It's this nice 3D structure. That's what gives it its tenacity. That and a couple of different things. Inside of this, we have what we call spore coat proteins. Inside of this, we have the cortex, and inside of this, we have the core. This is where we have all the goodies. So these would be all the various types of nucleic acids, like DNA, all the appropriate enzymes that's needed for replication and coming back to life. So all of those compounds and life necessities there. And the key is this thing doesn't have very much water. Each of these layers is pretty much water-free. And in the core, rather than having a lot of water, they have this other substance which is pretty nice because it allows it to hang out in these various conditions. It's called dipocholinic acid. So think of it kind of like an oil or a shortening that just kind of holds everything in animation in there so nothing can get out of control. So once we have formed our spores, they can go out and hang out forever, right? So we've all seen Jurassic Park. We get a piece of amber, create dinosaurs. That's not as far-fetched as you might think, actually. So spores are super tenacious. Because of this exosporium coat, resistant to heat, pressure, chemicals, radiation, etc. They've actually gone up, let them out into space, gathered them back up, brought them to Earth. Everything's worked out all right. And likewise, we've actually found them in amber in insects. These insects are millions and millions of years old. They actually go into the guts of the insects in amber, and they can pull these spores out of the guts. And I think the last one is something like 250 million years old. And they were able to pull these out and bring them back to life. Pretty crazy, huh? So they can hang out for a long, long time. Right now, some crazy stuff is going on with our climate change. For a few thousands of years, what was the last ice age? 6,000 years ago, 10,000 years ago, something like that. Our tundra has been frozen. It's called permafrost, right? Permafrost, permanently frozen. It's not permanently frozen anymore. So these organisms that were frozen 10,000 years ago are now thawing and coming back to life. And we're finding all sorts of stuff in the tundra right now. Viruses that are so large that you can see them almost with a microscope, which is unheard of. All sorts of crazy bacteria that have these spores, and the spores are coming back to life. So we're kind of bumping into these guys each and every day. Also, we've found them in supposedly spores in earth rocks. So they've been around during molten earth times. They are no longer viable, but we can see them as an old artifact. And meteorites, I don't know if that's correct, but it's close on. So they've suggested that there are these spore-like entities in Mars rocks, other meteorites from Mars that they have found. And I've talked about this before. That has given rise to the theory of panspermia. So basically that life has been blasted around the universe based on these spores traveling around. They land on habitable planets, come back to life. There you have it. We don't have to worry about how it all began. Makes it easy for us. Germination. So this is the growing out back to life. And we think of these living cells as vegetative cells. That means that they're living, replicating, using stuff for metabolism, all of that. This is a 3-step process. Activation, and this could be from many different things. Artificial heat, UV radiation, saturate them with nutrients. Then we get germination. And this can take minutes to hours, depending on the species that you're dealing with. And then eventually outgrowth. And these guys turn back into the living vegetative cell. So we've talked about all these cool prokaryotes with all their cell walls and their ultracellular mechanisms that are attached to the cell walls. So now we have to throw a spin on everything. There are these actually don't have real, I should put that next to that, real cell walls. So we'll call these prokaryotes without real cell walls. So we can develop these in lab using this enzyme called lysozyme that chews the majority of the cell wall away. So it digests the cell wall. And it digests it partially. So it leaves just a very, very small amount of it around. And we end up creating what's called a protoplast. These protoplasts are really fragile. And they only hang around for a certain amount of time. Then we have these others that hang out naturally without these cell walls. So one of these is called L-form bacteria. And these guys lose their cell wall in the presence of antibiotics that attack their cell wall. Take the antibiotic away, and they grow their normal cell wall back. Put them in the presence of antibiotics that attack their cell wall, and their way around it is just to kind of get rid of their normal cell wall. These are called L-form bacteria. Then we have all sorts of genera that are just naturally weird. So things like thermoplasma naturally don't have a real cell wall. And there are other ones as well. But we won't go into all of them. So recognize that even though we always try to show these bacteria as being a certain way, there are always these exceptions or outliers to it all. So when we're dealing with microbes, many times we just kind of show them as being these, like I said, sessile planktonic organisms that don't really interact with one another and all of that. But they really do. They have the ability to recognize what's going on in their environment and to adjust accordingly. And they're really good at it. And we can talk about these in a couple of different ways. The first way, we're just going to describe the general types of responses we see in microbes. Many of these you see in other organisms as well. And then we're going to talk about the process by which they carry out these various types of responses. So we think of these behavioral responses as a directed movement. Of organisms towards some sort of stimulus. We know these as taxis. And we have the various types of taxis. So chemotaxis. So, in this case, we're basically moving based on various types of chemicals, usually in solution, moving to, towards, or away from. Phototaxis, same thing. Movement based on light. So obviously those photosynthetic bacteria want to be in the light. Those that don't like light move away from it. Aerotaxis. Aerotaxis deal specifically with O2 concentrations. So we're moving towards or away from various O2 concentrations. A good example of this would be what we call anaerobes, obligate anaerobes. So oxygen is very, very lethal for an obligate anaerobe. They don't like oxygen, so they're going to do whatever they can to get away from it. And then, as I said before, we have a couple, both the magnetotaxis, which is kind of cool. We also have osmotaxis. Osmotaxis, in this case specifically, usually has to do with ionic concentrations in water, so sodium, salts, chlorines, that sort of thing. And then the magneto, which we've already talked about. Based upon that iron magnetite. So we ask ourselves, how do these little silly creatures take care of this process? They don't have any sort of nervous system, no sort of central brain that organizes what they do. Yet they give us all of these standard generalized functions that we see even up in higher eukaryotes. This process is called signal transduction. And the general basis behind this is that we receive some sort of environmental signal that's transduced through the cell membrane to the inside of the cell. And we end up with some sort of cellular reaction based upon that outside external signal. Now, there are many different types of signal transduction pathways. We're going to focus on specifically prokaryote because they're just so simple. If we get into eukaryote pathways, they're much more complex. Most of them are active, so they need either ATP or GTP to push this as an energy force. So we'll just kind of focus on these simple prokaryote processes. And if you're interested in these others, I can always talk about them later. So for prokaryotes, we have what's called a two-component regulatory system. It is comprised of what we call a membrane receptor, and many times we call these a membrane kinase receptor. And we'll talk about this. It has a bunch of secondary mediators. And these are called second messengers. And they're the main two components behind this. The receptor, which receives the signal, and the mediators that pretty much carry out the task. So when we draw this as a big blown up version of an E. coli or whatever it might be, of course my receptor is going to be completely out of proportion to what the bug is so that we can see it on the board. But these receptors have a transmembrane appeal to them. So the outside part, the membrane receptor, receives the stimulus, and the inside part is actually a kinase. And a kinase is an enzyme that adds phosphate groups. Phosphatases take off phosphate groups, kinases put them on. So we'll start with the signal. Once again, there's also another type of description of this. If you take a look in various texts, they might call it a receptor ligand interaction. The ligand is just any stimulus that binds a receptor. It's a more generalized term of describing this, but it's more or less the same. So here we have our stimulus, which we call the first messenger. let's just say that this receptor is a receptor for glucose, just to make things easy. So glucose comes down, binds the receptor. When this happens, the inside of this, the signal is transduced through the membrane, and the inside kinase basically activates second messengers. And it does it by adding these phosphate groups, so we'll just kind of put like a little red asterisk showing that it's been phosphatized. And then, of course, these guys go on and carry out whatever duty they might do. Usually they can turn genes on to do certain things, they can turn genes off, they make the cell produce more glucose receptors. In this case, the cellular response is, oh, shit, there's a whole bunch of glucose out in the medium. You need to make a whole bunch more of these receptors to bring the glucose in. That glucose goes away, the cellular response turns off, the glucose receptors go away. So for almost all of our taxis that we talked about, they have their own specific signal transduction system. Stimulus binds the receptor, transduces the message through the membrane, and then we get some sort of cellular reaction. So an example of this, which is really kind of cool, which we'll probably leave on today, is called quorum sensing. And we're going to come back to that in just a second. So for various types of these systems, so for instance, if we're dealing with O2 concentration, and that's the signal, it has its own signal transduction system called the ARC system, which is its own protein receptor and second messengers that are specific to this system. If we're dealing with nitrate respiration, or any sort of nitrate concentration, this is called the NAR system. Same thing, composed of receptors, second messengers that deal with dealing with nitrate. Same thing if we're dealing specifically with nitrogen itself, usually as an N2 compound. And so we're dealing with nitrogen concentration. That's the NTR system. So on and so forth. We have all these different systems. My favorite one is basically the quorum sensing system. It's its own unique system. And it's based upon this really great molecule that most organisms produce called acetylhomoserine lactose. Excuse me, lactone, also known as the AHL system. So quorum sensing doesn't sense an external stimulus like these other ones do. This guy is all about species concentration. So it can tell how many of its own species are around it at any given time by the amount of acetylhomoserine lactone that's produced. So let's give an example. Here we have our organism. It's got the receptors for AHL, but at the same time, it's also producing AHL and excreting it. Many questions have come up about how the AHL doesn't bind to its own receptors, and there's a specific reaction or mechanism that allows this to happen. So this AHL goes out into the environment. It binds to receptors on another organism, the AHL receptors. That has a second messenger effect. It makes this guy produce a ton of AHL. And we start to amplify this effect because the next bacteria starts to get the AHL to bind their receptors, they produce more AHL. And so the whole process is about recognizing the concentration of AHL in solution. If they recognize that there's a bunch of AHL in solution, they know there's a whole bunch of their own type of it. Why is this important? Well, strangely enough, these little buggers realize that they're so small and minuscule that if they carry out an action all by themselves, they're too small to have any effect. But if they can talk to their brethren and say, hey, let's all turn on our genes at once, they can have these great effects. So we see this in a few different pathways. Why do they do this? All about population size. Why would I carry something out if there's nothing around me to help me out? So I want to make sure there's plenty of my own little guys around so that when I do something, we can all do it together and have this really big effect. So, as we talked about toxins, salmonella in your guts just don't come in and go, oh, I'm going to bind to your epithelial and produce a bunch of toxin to make you sick, because they know that that one small amount of toxin is not going to do anything. So what they do is they come in and bind to the epithelial lining of your guts, your intestines, and they wait, and they replicate and replicate and replicate and replicate. And when the AHL gets to a certain level, they all turn on their toxin genes at once. And they all produce a crap load of toxin all at once. So that's one reason they do this. Same thing. There are specific systems that allow them to recombine their DNA. So if they're going to start to recombine DNA and there's no other organisms around to share their DNA with, it's kind of a futile attempt. So this is one of the ways they get this horizontal gene transfer. Infectivity. Many organisms wait to become really infectious and produce the sorts of symptoms you see until they have enough of their little buddies around to carry this out. And my favorite, bioluminescence. So you guys have probably heard about those little diatoms out in the ocean, and when the water moves, they all fluoresce green. You've probably seen them. Well, we also see this in bacteria. All of those organisms basically stole those genes from bacteria. Bacteria do the same thing. So many marine bacteria have bioluminescence. And a good example of this is this organism called Vibrio fischeri. And likewise, it waits till we get a whole bunch of its own light around, and then they all turn on their bioluminescence at once. And then it's super bright. So they've seen these Vibrio bioluminescent plumes from outer space, in the oceans and whatnot. They're super bright. If you've ever seen fishermen, Southeast Asian fishermen, a lot of them use these big green lights, these fluorescent green lights. Same thing, it draws all these organisms because these Vibrio people. And so, let's see. With that, we will start just a little bit of microbial growth and call it good for the day. We won't get too far into it. So, once again, we're going to talk about increase or decrease in cell numbers. And before we get into it too much, just recognize that microbes grow differently. So usually when we describe their growth, we're talking about batch culture in a broth. So that's different than when you streak them on a plate and grow them on an agar plate. We see different sorts of growth patterns. So when we talk about microbial growth, we're usually thinking about them in a broth kind of all together. And what we're going to do is just very quickly take a look at how these guys grow in the process of their growth curve. And it's pretty straightforward. If we were to take a big beaker of broth and inoculate it and look at it over time, and we compared it to the numbers per milliliter at any given time, we can see that these pretty much start at T0. So this is where we're basically taking an organism and inoculating into a broth. So just think about you using your loop, grabbing a little bit of something off a plate, sticking it into a broth. That's T0. What we see right off the bat is a pretty good lag time. Exponential growth, it tops off and then starts to decline over a period of time. So most of the time when we're talking about this is usually all within, say, about 45 minutes. And we would draw it out to months at the very end there, so kind of a slipping scale of time. This is called the lag phase. This is called the exponential phase. Stationary phase. And then the death phase. So when we hit lag time, boy, that guy's just making macronutrients, macromolecules, trying to get ready to replicate. So we see these guys just preparing to replicate, not very much. You could almost say that there's really no reproduction taking place. Exponential is where we basically see burst size. So we're basically doubling our population for every time unit. And then we get a stationary phase where everything kind of plateaus off. Growth and reproduction pretty much equal out death. Death phase, we've used up all of our nutrients. We've got tons of waste hanging out. And basically there aren't very many living cells. And we can say basically death far outweighs growth. And we'll come back and revisit it right there. We'll take about a 10-15 minute break, meet down in the lab.